Next up, our third speaker selected from our abstract contest is Sara Fallah, who's a doctoral candidate in the lab of Dr. Leah Cohen at the University of Toronto, focusing on novel antifungal research. I think I'm sensing a bit of a theme, and development. We talked a little bit about how this is a lacking area yesterday in terms of antifungal development, and it looks like we have some novel research happening that can potentially address. Today, she'll speak on identification and characterization of the Adamantyl diamine SQ109 as a repurposed inhibitor of squalene synthase. Squalene synthase in the fungal pathogen Candida albicans. Thanks, Sara. Come on up.
Perfect. So thank you so much for the introduction. I'm assuming the big green button is advancing. So fungal pathogens are major contributors to infectious disease, as we know. And broadly, we can categorize these infections into two main types. We have our superficial fungal infections, and we also have our systemic fungal infections. Now, our superficial infections infect over 1.5 billion people, and they can often cause recurring chronic infections that are make a significant impact on quality of life. And the majority of these superficial infections can be caused by the following dermatophyte genera. We have the trichophyton, the microsporum, and the epidermophyton, which cause infections like ringworm, or perhaps many of us are familiar with athlete's foot, hopefully not too familiar in the room. And on the other hand, systemic fungal infections cause over 6.6 million infections worldwide. 2.6 million deaths are attributed to these fungal infections, and the majority of these genera that are causing these systemic infections are genera like the Cryptococcus, the Aspergillus, as well as the Candida species, which I will be focusing on today, with the Candida albicans species dominating in terms of incidence across all patient groups. And Candida albicans is actually a commensal member of the human microbiota. And as we've seen throughout the theme of these past 2 days, unfortunately, the treatment of fungal infections is becoming more challenging due to resistance and the limited antifungal drug classes. So unlike the dozens of antibacterial classes that we have available, there are only 5 clinically available antifungal drug classes that are currently used to treat fungal infections in the clinic. And I want to focus on 2 in particular. The ergostro biosynthesis targeting antifungals, and the gold standard treatment for superficial fungal infections is currently the alinomines, and they target or help cure dermatophyte infections and other superficial infections, and they target the squalene epoxidase enzyme encoded by ERG1. And on the other hand, for systemic fungal infections, the antifungal drug that is currently the gold standard treatment are the azoles, with many of us perhaps recognizing fluconazole. And fluconazole, or the azoles, target 14-alpha demethylase enzyme, which is encoded by ERG11. But as you can imagine, unfortunately, both these classes are susceptible to resistance. So to combat this issue of limited antifungals, I resorted to phenotypically screening chemical libraries, as this is often a promising strategy to identify compounds with antifungal activity. And repurposing previously approved drugs for new indications can often expedite drug development as well as lower regulatory barriers and costs. And previously screening some of the other MMV libraries has successfully identified compounds with activity against diverse fungal pathogens. And at the time that I conducted this screen, the global health priority as well as the pandemic response box had yet to be screened against growth for Candida albicans. And both these libraries are comprised of structurally diverse compounds in various stages of drug synthesis and development. And screening of these 2 libraries led to the identification of a prioritized molecule called SQ109. And that's what I'm going to talk to you about today. And I'll split my time on how I identify and characterize this molecule, and then later we'll move on to therapeutic potential of SQ109. So I first determined minimum inhibitory concentration values of SQ109 using dose-response assays. And if you're not familiar with these beautiful green MIC graphs, we call them, I'll walk you through it. So green represents full growth, black represents complete inhibition of growth, and the readout is optical density, and you're normalizing your values to drug-free control wells. Here I'm reading at 48 hours after incubation at 30 degrees. And then I next looked at sidality, which is the ability of a compound to kill the fungus as opposed to just merely inhibiting growth. And here we're stamping from the liquid culture onto drug-free agar, and we're looking for the absence of colony formation where we see at the top there. I don't have a laser pointer. And in contrast to, for instance, the azole-fluconazole, where we know it's fungistatic, where we see the presence of colony growth where growth was previously absent in liquid culture. So we can see that SQ109 is fungicidal, which is a very promising result. I next looked at diverse fungal pathogens, so susceptibility to other fungal pathogens, and SQ109 had a pretty good portfolio with activity against all the pathogens shown on the screen here, minus Candida auris, which is formerly known as Candida auris. And I also looked at clinical resistant C. albicans isolates, and I found that SQ109 retained bioactivity against all the C. albicans isolates that I tested. So the benefit of using a repurposed drug really lies in that there can be a lot of literature on the compound. So interestingly, SQ109 has completed both phase 1 and 2 clinical trials for the treatment of Mycobacterium tuberculosis, and unfortunately there's no known homologue for its target in C. albicans, but I did some more literature searching, and I actually found that SQ109 inhibits the enzyme dehydrosqualene synthase from Staphylococcus aureus, and actually despite low sequence similarity between the two protein structures, I found that pairwise structural alignment and TM-align revealed a high degree of overlap between the superimposed predicted structures from AlphaFold of the Staphylococcus dehydrosqualene synthase and the C. albicans squalene synthase. So to explore squalene synthase as a target of SQ109 in Candida albicans, I leveraged the gene replacement and conditional expression library that we have in the Cowan lab. And this is a really cool genetic tool. Candida albicans is a diploid organism, if you didn't know, so we have 2 copies of each gene. And so this tool leveraged a heterozygous deletion mutant of C. albicans where the remaining allele is put under a tetracycline repressible promoter. So if you add doxycycline, you can get repression of your specific target gene. So I leveraged the ERG9 GRACE strain, where GRACE is the gene replacement and conditional expression library. And you can see where ERG9 encodes for squalene synthase. With the addition of doxycycline, you can actually see increased hypersensitivity to SQ109 compared to the absence of doxycycline, likely indicating that ERG9 is involved in SQ109's mechanism of action. And while this was compelling, we still wanted to gather additional evidence that this was indeed the target in Candida albicans. And through multiple experiments that unfortunately I won't have time to show to you today, we found that SQ109 treatment or genetic depletion of ERG9 resulted in vacuolar fragmentation, as well as depletion in lipid droplets, which is consistent with literature. And as a more direct approach, I'll show you this, but to examine the effect of SQ109 on sterile biosynthesis, we employed both GCMS and LCMS to quantify the abundance of sterile intermediates following SQ109 treatment. So if you're predicting here that on my really simplified ergosterol biosynthesis pathway here, if we're predicting that SQ109 is targeting ERG9 or squalene synthase, we would expect an accumulation in the precursor, farnesyl pyrophosphate, and then a depletion in other downstream sterols that we quantified. And that's exactly what we saw. With LCMS, we quantified farnesyl pyrophosphate, and we saw that with the treatment of SQ109, we saw an increase in accumulation, and in contrast, other downstream sterols like squalene, lanosterol, and ergosterol depleted upon SQ109 treatment. And once again, the genetic repression with this ERG9 GRACE strain resulted in the same results. So we saw accumulation in farnesyl pyrophosphate and decrease in squalene, lanosterol, and ergosterol, confirming that SQ109 inhibits the squalene synthase enzyme encoded by ERG9 and C. albicans. So moving on to assessing the therapeutic potential of SQ109 as my final arc of my talk. And I want to focus on this ability that Candida albicans has to transition from yeast to filamentous form. And this happens in various filament-inducing cues, we call them, such as high temperature, the presence of serum, or internalization by macrophages. And so I wanted to see if SQ109 at concentrations that did not impact growth would inhibit this transition, because that would suggest that it's a promising antifungal molecule. And so I tested SQ109 in high temperature in the presence of serum, and we can see, hopefully not super well from these grainy images, but maybe you could turn your head to the side very briefly, and you can see that with SQ109 treatment, we see these yeast locked in yeast form, in contrast to the untreated control, where we see these really long filaments in these filament-inducing cues. And I also wanted to look at co-culture models, so this internalization by macrophages as well, to just see what that would look like. And I actually found that the block in filamentation was also true in co-culture with mouse monocyte macrophages. So here I'm infecting these mouse macrophages with Candida, and Candida is highlighted in the green here with the antibody, and then DAPI is stating the nuclei of the macrophages. And we can see that, again, Candida is locked in its yeast form, in contrast to this filamentous form that are piercing through the macrophages. And this block in filamentation in co-culture actually was sufficient to improve host viability through fungal clearance. And as we've seen previously throughout this talk, these phenotypes were true for the genetic repression of ERG9 in the conditional repression mutant. So Kirsten introduced this really beautifully, but I wanted to talk also about C. albicans biofilms, because they're inherently resistant to antifungals, and the host has a really difficult time clearing this from an infection. So they pose a really significant clinical challenge. So I wanted to see if SQ109 also had this ability to prevent biofilm formation, as filamentation and biofilm formation are often linked. So here I'm showing that in high concentrations with SQ109 treatment, we see a prevention in biofilm formation, in contrast to the known azole control, fluconazole, you can see right here, is not inhibiting biofilm prevention. And this is exactly what we saw when we treated with SQ109 on already mature C. albicans biofilms. So we also saw that block with SQ109 treatment. And we wanted to apply this in a more clinically relevant context. So we collaborated with Dr. David Andes at the University of Wisconsin, and we used a rat model of catheter-associated candidiasis. And this mimics a common complication of central venous catheters in patients. And so here what we found was, again, very beautifully, SQ109 resulted in a decrease in CFUs relative to the untreated control, which was a really promising preclinical result. So while SQ109 displayed significant potential as a broad-spectrum antifungal, unfortunately, we were unable to observe any significant therapeutic effects in a systemic model of candidiasis. So we turned to an alternate model of infection. We looked at a topical infection model, and we tested the efficacy of SQ109 in a mouse dermatomycosis infection of trichophyton integraphytes. And here we used mouse immunosuppressed mice, and we infected them with trichophyton integraphytes. We allowed the infection to establish for 4 days, and at 4 days, we treated for 7 days with twice daily, either with our untreated vehicle control or 2% SQ109 in cream formulation. And then we plated for CFUs. And from these images, it's a little hard to tell, but you can see with the SQ109 treatment, you can see that the mouse skin is less infected with the white patches of fungus, and it's less irritated overall, especially in comparison to the untreated control. And this is further exacerbated with the CFUs that I'm showing. So 2% SQ109 actually resulted in a limit of detection. So we weren't able to detect CFUs because it worked quite well, in contrast to the vehicle and untreated control, overall showing that SQ109 has extremely promising results in this preclinical model. So to summarize, I hope that I've shown you how screening previously approved drugs for new indications can often help identify molecules with antifungal activity. SQ109 inhibits squalene synthase in the fungal pathogen C. albicans, and it also shows promising results in inhibition of filamentation and biofilm formation, both in vitro and in vivo. And finally, we've shown promising results in a mouse model of dermatomycosis. So I'd like to thank the organizers for having me, my supervisor Leah in the lab, and my collaborators as well as the funding sources, and I'm happy to take any questions if there's time.